Hi everyone, welcome back to the Immunology series. Now in this lecture, we will begin to have a brief overview of the basic principle of autoimmune that are common to most autoimmune diseases. Now, however, we do not cover any specific autoimmune diseases in detail in this lecture. So let's get started. Here are the lecture objectives. We will compare and contrast the central and peripheral mechanisms of self-tolerance for T cells and B cells. Look at what contributes to autoimmune diseases and how it sometimes is quite similar to hypersensitivity reactions. We will wrap up with an example of how infection can trigger autoimmunity. Autoimmune disease basically is the body turns on itself. Now, there are more than 50 different diseases classified as autoimmune. Now, the prevalence of disease is like allergy. It has higher prevalence or incidences in industrialized countries. And as a group, there's about 5% in the industrialized countries. Now, the root cause of autoimmune disorders is some type of failure of the mechanisms for self-tolerance. There are two types of self-tolerance mechanisms, central tolerance and peripheral tolerance. The chief mechanism of T cell tolerance is the deletion of self-reactive T cells in the thymus, and we call that negative selection. The same process also happens for B cells in the bone marrow. Now, however, it is not a foolproof process, and cells that are mildly reactive to self-antigen may escape into the circulation. Now, so it is important to have peripheral mechanism to maintain self-tolerance. In the periphery, some T cells are referred to as ignorant. Now, they ignore the antigen even though specific to it because the level or the amount of antigen present below the threshold for reactions and activation. Now, our bodies also have specific anatomical barriers to prevent T cells from accessing, and such as the CNS, anterior chamber of the eye, endocrine glands, and testes. Now, these physical barriers create so-called immunological privilege sites. Now, these barriers also prevent lymphatic drainage. Some T cells also express something called CTLA4, cytotoxic T lymphocyte associated protein 4, which can be a receptor or can be in a soluble form. Now, this can bind to B7 on antigen presenting cells with a higher affinity than the co-stimulation receptor CD28. Now, in animal study, it has been shown that the blockade of CTLA4 accelerates the progression of autoimmune diabetes in mice, so it plays a role in the autoimmune development. Another mechanism to maintain peripheral tolerance is called energy, starting with an A. Now, it means cells are in sort of a suspended animation where the lymphocytes are non-responsive to specific antigens, but it is still capable. Now, it is due to the lack of co-stimulation required for antigen presentation. Another way to induce T cell tolerance is to have a suppression by the regulatory T cells, or commonly abbreviated as Treg cell. Now, this is our first time talking about this different population of T cells. These T cells have both CD25 and CD4 on their surface, but unlike other T cells, the Treg cells are specific to interact with the self-antigen or the self-peptide. Here you are looking at an illustration of how the T regulatory cells interact with the antigen-presenting cell. The antigen-presenting cells present a self-peptide, and there is a CD4 T cells that interact with the self-peptide. Now, this Treg cell releases different types of cytokines to suppress the activity of CD4 T cells that are self-reacting, including interleukin 10, which can inhibit the cytokine release or synthesis, Another cytokine is the TGF-beta, which can inhibit the mitosis and promote apoptosis of those self-reacting cells. And lastly, we have CTLA4, which can act as a binder to inhibit or to bind and block the interactions on the B7 marker. So these are the mechanisms for Treg cells to suppress the proliferation of naive T cells responding to self-antigens presented on the same antigen-presenting cell. B-cell development is depending on a network of non-lymphoid stromal cells in the bone marrow, which provide environment and growth factors for B-cells to develop. There are two fate-determining checkpoints during B-cell development in the bone marrow. Both checkpoints are marked by whether a functional receptor is made, which is a test of whether a functional heavy chain which is at the first checkpoint, or a functional light chain at the second checkpoint has been produced. Now, cells that failed either of these checkpoints die by apoptosis. Immature B cells that are not specific for self-antigen in the bone marrow mature further to express IgM and IgD and leave the bone marrow. Immature B cells specific for a self-antigen on bone marrow cells are retained in the bone marrow and may undergo a reprogramming step called receptor editing. In fact, many self-reactive B cells are rescued by receptor editing process, which changes their antigen specificity. Now, binding to a self-antigen in the bone marrow causes the immature B cell to continue to rearrange its light chain locus on the gene. This deletes the existing self-reactive light chain locus, and rearrangement may also produce a new light chain to give rise to immunoglobulins that is not specific for self-antigen. Rearrangement is successful and generates non-self-reactive immunoglobulin receptors. The B cells continues its development, or if no rearrangement are possible and the Ig receptor is still self-reactive, the B cell will undergo apoptosis. So in other words, B cells do get a couple chances before they are programmed to have cell death. In the peripheral, B cells can also undergo energy when they encounter autoantigen or self-antigen. Now, when the B cells bind to it, instead of activating, the massive binding due to overabundance of self-antigen will cross-link multiple bound IgMs on the cell surface. This excessive binding to self-antigen sends an alarm and causes cell cycle arrest and stop the development. Afterward, surface IgM receptors are sequestered or internalized in internal cellular compartments where they cannot reach and react to antigens on the cell surface. This leads to decreased expression of Ig receptor on its surface. Now, when we look at all autoimmune diseases, here is a common theme that is based on the breakdown of self-tolerance, leading to sustained and continuing action by the adaptive immune system against normal and healthy tissue, leading to chronic inflammation and infiltration of lymphocytes. Now, the key is that there is a breakdown in the T cell or B cells tolerance that is particularly important because without the CD4 T cells help to activate the B cells, subsequent differentiation into plasma cells and secretions of antibodies, the response would not be sustained or amplified. There are some endogenous factors that are involved in autoimmune diseases, and sometimes they are linked to genetics, such as having a special type of HLA2 or MHC class 2, which is responsible for presenting the peptide to CD4 T cells. Some types of these MHCs are more prone to presenting self-peptide to CD4 T cells, and also there could be exogenous triggering factors such as infections or physical trauma or chemical insult. After the breakdown of self-tolerance, people will develop autoimmune diseases, which is a chronic state of healthy tissue being attacked by host immune system. And depending on the specific autoimmune disease, the self-attacking mechanism can be either by autoantibodies and or overreactive T cells. It is very common that the effectors of autoimmunity duplicate hypersensitivity reactions, specifically type 2, 3, and 4 hypersensitivities. Now remember from our last lecture, type 2 and type 3 hypersensitivities are characterized by having IgG involvements. We observed similar phenomena in Graves' disease, in which self-reactive IgG binds to TSH receptors and causes the overproduction of thyroid hormone. And in lupus, autoantibodies to common antigens attack multiple organs, form complexes that deposit in the walls of blood vessels throughout the body, and lead to systemic inflammation. Where overreactive T-cell mediated inflammatory responses result in tissue elimination in diseases such as multiple sclerosis, inflammatory bowel disease, rheumatoid arthritis, and psoriasis. Another autoimmune disease contributing factor is the accumulation of Th17 cells. The Th17 cell is a subpopulation of CD4 helper T cell. Th17 can secrete IL-17, which is a pro-inflammatory interleukin that can trigger even more cytokine release by the fibroblasts, epithelial cells, and also keratinocytes that underlies the skin. Th17 cell can also secrete IL-23, which has a role in promoting T cell differentiation in diseases such as Crohn's disease, rheumatoid arthritis, psoriasis, or allergic rhinitis. Now, in these diseases, Th17 cells accumulations has been associated, and because IL-17 and IL-23 play such an important role in autoimmune disease development, there are specific monoclonal antibody treatments that target these cytokines. Here we are going to look at how infections can sometimes trigger autoimmunity through the mechanism called molecular mimicry. One of the potential triggers for autoimmunity is infection through a mechanism called molecular mimicry. In molecular mimicry, there is cross-reactivity between self-peptide antigens and pathogen peptide antigens. When proteins get broken down into a single small piece of peptide that is a couple of amino acids in length, sometimes our native peptide may have very high similarity to those foreign peptides. And these similar peptides are called homologous native peptides. And because they are so similar, and sometimes our immune system can mistakenly think they are from pathogens and start to react to them, for example, in a condition called rheumatic fever. Here is an example of how rheumatic fever develops. Now, the adaptive immune system mounted an immune response to streptococcal infection and generated antibodies that can react to the bacterial cell components. But some antibodies are specific to the bacteria. The similarity between bacterial antigen and hard tissue self-peptide at the small peptide level makes them also attack hard tissues. This can lead to activations of complement, inflammation, and in some cases are enough damage to the heart that can lead to heart failure. Molecular mimicry can also happen from the T cell perspective. The similarity between the self-peptide and the pathogen-derived peptide stimulates T cell responses. In the figure, we see the whole red dot is a pathogen peptide, and the self-peptide is partially similar. But after T cells are activated by these similar-looking pathogen peptides, when they see the self-peptide, it mistakenly recognizes it as pathogenic and activates macrophages and causing inflammation and autoimmune disease development.